Professor White. Well, the first thing I want to do is apologize for that picture that's posted on the cover of the booklet, because as you can see, that's a gross misrepresentation of my current state of oxidation. Second thing I'd like to thank is Professor Hashmi and the organizers to invite me to participate in this Congress. And this is my third trip to Riyadh, and each time it's a great opportunity to renew friendships and to share some ideas about these disorders that we're treating. So here are my disclosures, none of which really pertain to the presentations today. So as we heard last night, allogeneic bone marrow transplantation is really the gold standard as a curative therapy for the hemoglobin disorders. On the left panel are data from a group of patients treated in China recently reported, and then on the right are data from the CIBMTR and for the European bone marrow transplant registries in sickle cell disease. And what they both show is the results are very good when there's an HLA identical sibling donor and the transplant is performed when in young patients who are in good shape. The curves are less good when a mismatched or alternate donor is utilized in patients with advanced disease. So it suggests that if we could utilize a treatment that uses autologous cells and rescues from the hemoglobinopathy, this might have a benefit in avoiding the problems of graft versus host disease and graft failure, which are the leading causes of failure after allogeneic transplantation. So since only a small fraction of patients will have an HLA identical sibling donor and a similar number will have well-matched unrelated donor, these are patients in the United States. Even if I wanted to treat every patient, I could only treat about a third by allogeneic transplantation. And in addition, because of this problem of graft versus host disease and transplant-related morbidity and mortality, clinicians and families, patients often decline this as a therapeutic option. So transplant, at least in our country, is largely restricted to children and applied very sparingly. So the first question is, can we modify autologous cells for clinical benefit? And then the second important barrier, and perhaps even more important, can we ensure equitable access to these novel curative therapies as they're developed, where the diseases are endemic worldwide, such as South Asia, Sub-Saharan Africa, and the Middle East. So these are some broad categories of genomic or gene therapies. The first category that I'll spend some time reviewing is gene addition therapy using an anti-sickling beta globin or a fetal globin, rescue globin, and these can be used interchangeably in thalassemia and sickle cell disease. I'm not going to talk at all about gene editing to induce fetal hemoglobin, although that's an area of active research targeting the regulator BCL11A to reinduce fetal hemoglobin in adult stem cells. And then gene editing to correct the sickle or thalassemia mutation directly. So this is not yet in clinical trials, but has some exciting advantages. And then which is not ready for prime time is our in vivo gene editing techniques to correct mutation. But really in the future, ideally this is where we would go to get away from ex vivo manipulations and the problems therein. So this is the vector developed by Philippe Labouche and later licensed by Bluebird Bio. It's a lentiglobin vector, BB305. It contains erythroid control elements ensuring expression only in erythroid-specific cells. It has a CMV promoter driving the packaging signal, so very high titers can be developed, and it's self-inactivating, so replication-competent lentivirus can't be developed. It contains a single amino acid change that we call T87Q. There's a swap of threonine for glutamine because it has anti-sickle polymerization activity. And because of the single amino acid change, this hemoglobin can be monitored by HPLC post-drug product infusion. So it's a very nice way to follow the impact of the therapy, and if a level of 30% or higher can be reached in sickle cell disease, it should have significant disease-modifying effect. So this is how patients are carried through the clinical trial. in thalassemia, hematopoietic stem cells are mobilized by GCSF and plurixa-4. These are shipped to a central manufacturing facility where they're transduced with an lentiviral vector, cryopreserved, and if they meet release criteria, they're shipped back to the clinical center where patients receive a single conditioning drug, busulfan, that accomplishes myeloablation, and then they're followed for 2 years post-infusion for outcomes. There's an option of participating in a 15-year long-term follow-up study. And illustrated on the lower right-hand panel is the team in Oakland that carries out all the logistics of these procedures. So these data were published nearly 2 years ago now in the New England Journal of Medicine, where the HGB-204 North Star study was performed, enrolled 18 patients who currently have a median follow-up of 32 months. And all of these patients have elected to enroll in the long-term follow-up study. So the primary objective was to assess the safety and efficacy of the lentiglobin gene therapy in transfusion-dependent thalassemia. So these are 2 groups of patients that were part of that 18-patient cohort. The upper panel are patients who had a non-beta 0 genotype. So these are individuals, adults who have transfusion-dependent thalassemia, but generally start transfusions later in life because they make some small amount of endogenous beta globin. The lower panel is a more severe genotype termed beta 0, where no beta globin is made after the fetal globin is turned off at birth, and so they require more expression of the gene therapy hemoglobin to achieve transfusion dependence. So what's shown in light blue in these swim lane plots is the time in months during which transfusions, red blood cell transfusions, were administered after the myeloablation. And then the dark blue indicates the time in months since the last transfusion was administered. So 9 of the 10 non-beta 0 genotype patients have stopped transfusions, and 50%, 4 of 8 of the more severe genotype also have stopped transfusions. So really a remarkable outcome in that initial study. The next iteration, called North Star 2, involved an important improvement in the manufacturing, a refined manufacturing process that significantly increased the vector copy number. And that's important because the higher the vector copy number, the higher the level of the gene therapy hemoglobin, the T87Q. And so that's illustrated in this panel on the right. So in the 204 study that enrolled 18 patients, the vector copy number on average was 0.7, and that improved to 3.1. And that translated into, instead of having 30%, 31% of the CD34 positive hematopoietic stem cells marked by the lentiviral vector, to 82%. And that's an important factor. So in both studies, patients received myeloablative dosing busulfan, and the time to neutrophil and platelet engraftment is shown in that lower right-hand panel. So before I go on to show the results, I want to summarize the adverse events and the toxicity of this approach. All the patients had at least one grade 1 or higher adverse event, and 20 to 34% of the thalassemia and sickle cell disease recipients had a serious adverse event. Most of these were related to the conditioning, so febrile neutropenia, mucositis were the most common ones, but there was this important problem of veno-occlusive disease of the liver that happened in nearly 10% of the thalassemia recipients, treated by defibrotide, which is a way to manage this serious complication, but it was a grade 3 or grade 4 complication. Luckily, all the patients recovered. And of interest, it just happened in thalassemia, probably having to do with the iron loading that occurs in that disease early on in life. It didn't happen in sickle cell disease. The other important serious adverse event that occurred in a sickle cell disease patient was myelodysplastic syndrome monosomy 7 that happened in one of the sickle cell disease patients that progressed to acute leukemia, treated by an HLA haploidentical transplant. So this was not genotoxicity, so testing of the hematopoietic stem cells, the leukemia stem cells showed no vector in those cells, but it was probably related to the busulfan conditioning, which is an important risk factor for this therapy. So this shows in the current North Star 2 study, the gene therapy hemoglobin in pink, that's the T87Q, and the total hemoglobin in blue. over time in months post-infusion. And in this study, the peak hemoglobin was achieved by 6 months post-infusion, and the mean T-to-7Q level was 8.7 to 9. So instead of 5 grams per deciliter in the initial study, now it's up to 8 to 9 grams per deciliter, which is translated into a high rate of transfusion independence. So in these 20 patients, 18 achieved transfusion independence in the first 3 months post-infusion, shown by this swim plot. And the patients in green are actually children less than 12 years of age who also participated in this study. So this remarkable result was the basis for the EMA licensing and approval of the drug for this indication in non-beta zero and beta plus beta zero genotypes in Europe. A parallel study targeted the beta zero genotypes. So here are data from 11 patients, and the hemoglobin at last visit is shown on the right, ranged from 7.8 to up to 14 grams per deciliter. And as expected, this is a higher bar to achieve, but still 9 of these 11 patients have also transitioned to transfusion independence, have stopped their red blood cell transfusions post-drug product infusion, although in some cases it takes up to 9 months post-infusion to reach that threshold. So what's the impact of transfusion independence and having a gene therapy hemoglobin that's 8 to 9 grams per deciliter? Well, the markers of dyserythropoiesis are markedly improved. So the markers in the HGB-207, the North Star 2 study, are the soluble transferrin receptor level, the absolute reticulocyte count, and looking at the marrow exam at 3 months post-infusion, the MDE ratio, all of which improved or nearly normalized in the patients in blue who had a suitable vector copy number in the peripheral blood. And in the beta zero genotype patients in the HGB-212 study shown in the bottom panel, the soluble transferrin receptor also is improving in the patients that have a suitable vector copy number post-infusion. So this all looks very encouraging. I'm going to shift now to the story with sickle cell disease using the same vector, but remember it has an anti-sickling amino acid change. So this study journey is a little bit different. So G-CSF can't be used in sickle cell disease because it causes leukocytosis that can precipitate very serious vaso-occlusive complications. So initially marrow was used as the source of stem cells, but because of problems marking hematopoietic stem cells long-term, this evolved to use plerixafor as a single mobilization agent. So in the group C, using these optimized methods, all the patients received plerixafor, mobilized peripheral blood, received transfusions to reduce the hemoglobin S less than 30%, pre-collection, and then applied that refined manufacturing process to raise the vector copy number. So focusing in this talk on the group C patients, 49 patients have been enrolled, and 17 as of this data cut have had the lentoglobin drug product infused. So what are results in those 17 patients? So these are the baseline characteristics. All 25 of the patients enrolled to date have had frequent vaso-occlusive episodes, a median of 4 hospitalizations per year. And then in addition, the smaller number of patients have had acute chest syndrome, a history of stroke, and elevated tricuspid regurgitant jet velocity as a risk factor for sudden death. So this is a group of quite ill young adults and adolescents. Patients required a median of 2 mobilization cycles to mobilize and collect a sufficient number of hematopoietic stem cells for a drug product manufacturing. All the patients received myeloablative busulfan. The time to neutrophil and platelet engraftment is shown, 20 and 28 days, and patients were hospitalized for a median of 5 weeks. And in this, using these peripheral blood stem cells and then transduced with refined methodology, an average vector copy number of 3.6 was achieved, which represents marking in 80% of the stem cells. So in the group C patients, this shows the percentage of lentiviral mark cells in the peripheral blood in months post-infusion, and they range from 30% up to 80%. And this contrasts to the patients who received bone marrow as the stem cell source, where fewer than 10% of the cells were transduced. So a significant improvement in the transduction frequency and the stability of the vector copy number over time. So what did this level of marking translate in the blood? So follow the red bar over time through 21 months post-infusion. That's the T-DA7Q anti-sickling hemoglobin. And by this 12-month time frame, the hemoglobin S level in green was approaching 50%, and at the last time point, it's actually less than 50%. So that really looks like sickle cell trait. So really the question is that a mixture of cells that contain 100% S in 40% of the cases and 100% TD7Q and 60%, or is this pancellular distribution as occurs in sickle cell trait, which is an important distinction. So one of the predictors is shown in this slide. These are markers of hemolysis, reticulocyte count, lactate dehydrogenase level, and total bilirubin, all of which nearly normalized post-infusion. So it suggests that these transduced cells are behaving more like a sickle cell trait than a mixture of cells that are likely to hemolyze and those that are not. But this was really proved nicely by a new assay. So this is a single-cell Western blot assay using a cocktail of 3 different antibodies. So there's an antibody for the sickle hemoglobin, an antibody for hemoglobin A, and then an antibody for the hemoglobin A TD7Q. So you mix them all together, and then you ask the question, does it look like this? So this is a wild-type individual, so only stains with hemoglobin A. This is a patient with homozygous sickle cell anemia, so only stains with the sickle antibody. And this is a patient, a schematic of a patient with sickle cell anemia who's receiving red blood cell transfusions. And so you see 2 types of cells, one that stain only with hemoglobin A and one that stain only with hemoglobin S. And this is in distinction to sickle cell trait, where you see this stippled pattern, and then after the lentiglobin infusion, the prediction is that this would mimic more closely sickle cell trait than this. So in fact, that's exactly what was seen over time post-infusion. So initially, post-infusion, you see this mixture of red cells that only contain the transfused hemoglobin A and the sickle hemoglobin, and as the transfusions are stopped, you see this replacement with the stippled pattern where most of the cells, almost all the cells, contain at least some of the anti-sickling hemoglobin. So what impact does that have clinically? Well, that's just showed again by another swimmer's plot, and this shows severe vaso-occlusive events resulting in hospitalization before drug product infusion, and each event is depicted by one of these diamonds. And then after infusion in yellow, you see, and to a large extent, no diamonds. So this was a single vaso-occlusive episode that was non-serious, treated at home, in one patient about 3.5 months post-infusion. So a profound improvement in the clinical expression of the sickle cell disease, as you might expect if this truly mimics sickle cell trait. So a very exciting point in the development of this new gene therapy product. So I'd like to finish with some work we're doing locally in Oakland. This is a disease gene editing project led by our team in Oakland that focuses on the hematology of the gene editing, and then a collaboration with a group at University of California, Berkeley, Jennifer Dadna's group, who provides the expertise in CRISPR-Cas9, and then Don Cohn at University of California, Los Angeles, who provides the expertise in manufacturing. So we have expertise in sickle cell disease and the hematology of the disease, gene editing, and then manufacturing. So the basis for attempting this has to do, it relies on this observation of stable and mixed chimers, and that happens about 5 to 10% of the time after an allogeneic transplantation for sickle cell disease and thalassemia, where it's observed that as few as 20% of the donor cells is sufficient to ameliorate the sickle cell disease and thalassemia. And this has to do with the improved survival both in the circulation and also amelioration of the ineffective erythropoiesis that occurs in both disorders by virtue of having a fraction of donor cells in the marrow. And so it indicates to us that correction of at least one sickle mutation in at least 20% of the engrafted stem cells should be curative using the CRISPR-Cas9 approach. So this is a wonky schematic of what we've designed. So the sequence shown is the single-stranded oligo donor, and this is the template for homology-directed repair. And then the guide RNA is shown in red. So this is all anchored. The Cas9 is anchored at this PAM site. And what we do is we introduce a mutation at the PAM site so that once cleavage has occurred, it can't occur again using the same molecule. And so at the cleavage site, that's where the HDR, the homology-directed repair, begins. So most of the time, there's an insertion deletion that occurs, so HDR does not occur, and that essentially introduces a thalassemia mutation. So addition or deletion of a single base pair knocks out the beta globin gene, so that's bad. The other thing that could happen is nothing would happen at all and you retain the sickle allele. That's the second bad thing that can happen. And the only good thing that can happen is that HDR will occur. It will extend from the cleavage site all the way to the sickle mutation where we'll restore the glutamic acid wild type sequence. A small number of time HDR begins, the homology direct repair begins, and the strand elongation falls off before it reaches the sickle allele. So that's an important, but it does change the PAM mutation. It doesn't insert this PAM mutation, and we can use that as a control in just a second. So remember these small details about this figure. So what we've done is we've taken discard hematopoietic stem cells from a sickle cell disease patient. We've performed the manufacturing protocol and corrected the sickle allele. These hematopoietic stem cells are then injected into an immunodeficient mouse, NSGBW, and then at 20 weeks, at which time you would you expect engraftment of the human stem cells in this xenotransplanted mouse. We can then do genotyping to assess in each of these mice, there are about 43 that were analyzed, what was the frequency of HDR, where the sickle allele is correction, and what was the frequency of introducing this thalassemia allele by insertion deletion. So the good news is that we corrected on average 23% of the alleles, which by the mixed chimerism data would suggest is sufficient for a cure. The bad news is that we introduced 65% of these null alleles, which will induce a thalassemia phenotype if not balanced by correction. So the question we ask is that sufficient to have a clinical effect? Well, you won't really know until you conduct the clinical trial, but we did get some insight by looking at BFUE. These are erythroid progenitor colonies from the marrows of these xenografted mice. And so this is busy, but this is the most important slide I want to show you about this project. So these are the unedited controls, and looking at marrow, CD34 positive stem cells, or glycophorin expressing erythroid progenitors, there's no difference in their representation in the unedited cells. And then remember those HDR events that fell off the template before it reached the sickle allele? We call that PAM only, homology-directed repair. Again, no enrichment whatsoever because the sickle allele wasn't corrected in the erythroid lineage. But look what happens when the sickle allele is corrected by homology-directed repair, comparing the erythroid progenitors to the stem cells in unfractionated marrow. Well, it reaches nearly 50% representation, which looks to us a lot like sickle cell trait. And the out-of-frame beta 0 alleles that we've induced by the insertion deletions, these really fall out in the erythroid lineage, so they don't contribute to definitive erythropoiesis and red cells that eventually will find their way into circulation. And then if these are in frame, there's a little bit of enrichment as well. So it suggests to us that that level of correction may be sufficient for a strong clinical effect, and it has given us encouragement to proceed with the, I guess, one of them now 500 or 600 INDs that will be submitted to the FDA for consideration. So in summary, I've tried to introduce the notion that there's probably an ideal cell therapy profile that should protect from hemoglobinopathy-related complications, both clinical and subclinical. There should be an acceptable toxicity profile in the short term and the long term. It should be accessible and available to most patients where they live, and safe in children and adults. And eventually, I think we're going to need to perform comparative trials that show evidence of benefit of the curative therapy compared to the standard therapies. And then, of course, I'd like to acknowledge all the co-investigators. These are the sites where the thalassemia studies were done with the Bluebird Bio-sponsored vector. These are the sites that participated in the sickle cell disease trials. And then this acknowledges the funding agencies that enabled us to do the work. Thanks very much.
Thank you very much, Dr. Walter. We can take one question because of the time.
Yeah, thank you very much, Professor Walter. In terms of cost, so how does somebody or a panel or a ministry or a country on a nationwide scale select patients or define criteria in a way that is cost effective and doesn't break the bank?
I have no idea. I think that's really an important question, and there are some novel repayment schemes that are being developed, but I think that's why it's important to have both industry-sponsored and academic-sponsored projects like the ones I've described, because our approach in the end, if this were successful and did show a benefit, would be to keep the manufacturing cost at cost so that it really could be accessible to patients. Now, there's not really a model for how to do that, but that's the lofty aim that we have for our own particular project.
Can I ask one other question?
Because of the sake of the time, we are running out, maybe later during the breaks. Thank you, Dr. Walter, for this interesting talk. Next